Just to make a quick, very quick resume of Mahdi's career. He has done his PhD in Roscoff in France, and then he has been working in UK, in Plymouth. He's been working then in the US, in Indiana, and then he went back to Europe, working at Oxford. He moved between the departments in Oxford. He was in the plant laboratory before, and now he's working in a science department. And we can congratulate Mahdi, because he will start a new position in Rimouski in Canada this summer as a professor. So congratulations to you, Mahdi. I will leave you now, Mahdi, for his talk. Just please, Mahdi, could you share a full screen? And okay. It's up to you now.
Okay. First, thank you, Baptiste. And also, good morning, good evening, good afternoon, everybody. I don't know on which timeline we are. But I'm very honored to present you some aspect of my work on coccolithophores. So I have been working for quite a long time on the genetic aspects of coccolithophores, especially related to diversity and speciation. So the title of my talk right now is on the origin of new coccolithophore species. It's not that original. It's a bit redundant with something that had been provided a long time ago by Charles Darwin with the origin of species by means of natural selection. But that question has never been really solved or completely assessed on most marine phytoplankton species. And one of that existential question is how new phytoplankton species emerge in open ocean. And that question is still something that can be applied to any type of organism in the living realm. There are some species concepts that we first need to define, such as the biological species concept, the ecological species concepts, the evolutionary, the morphological, and the phylogenetic. So all those species concepts are based on a different approach or different aspect of an organism. It can be based on living organisms, but it can be also based on fossils, trace of those fingerprint of living organisms. So one of the main aspects I've been interested in is how to combine those species concepts on coccolithophores, knowing that we have a huge fossil record and quite a lot of work that have been done on the taxonomy of those organisms. And what we know so far in the living coccolithophores. So linking the biological concept with the morphological concept is something that had been very trivial, as you can see here, especially when we use DNA to accompany, to refine those concepts. We can go into the concept of evolutionary species, or the concept of or the phylogenetic concept. Adding the DNA part, the genetic part is showing how speciation is mostly working in a way as we have usually a mutation of an accumulation of mutations between different parts of an ancestral species that can be split into two, for example. And due to selection or drift, we are ending up with different phenotypes genetic divergence and therefore different species. Those process of speciation can be also defined by some spatial aspects, such as the allopatry, the peripatry, the parapatry, or the sympatry. When you start with your original population, you may have different initial steps of speciation. The most common model for speciation is the implementation of a barrier that will later on isolate two populations, and through time you will have those accumulation of mutations. Those two populations will also evolve towards reproductive isolation, so they will be no longer able to reproduce. So again, we are dealing with a biological species concept. If they are not able to reproduce, they are therefore two different species. After more time, those species can co-occur in similar ranges, but as long as they are not reproducing, they are two different species. The other, let's say, the most extreme pattern of the most extreme mode of speciation is called the sympatric mode, where the initial step of speciation is coming from genetic polymorphism within a population. So there is no spatial separation between those populations. It's really occurring within the population, and the reproductive isolation can occur as well in such case. That has been seen in many bacteria, for example. The peripatric and the parapatric are more or less some kind of the gray shade of those two extreme mode of speciation. So again, I'm coming back to that question, how new phytoplankton species emerge in open ocean? The open ocean is quite vast, and almost all currents are interconnected. But when we look at how the chlorophyll is distributed on this map, we can see some structuring patterns. So maybe it's not that simple. There might be some places where we may have some sympatric speciation, and some other where we'll have some allopatric speciation. But there is a good chance as well that we will have all the other shade of model within the range of the phytoplankton. Also, when we're talking about speciation in phytoplankton, when we think about most of the species that have been studied on the terrestrial realm, a lot of them have left a fossil record, and not a lot of phytoplankton have left such imprint in the sediments. So there are some dinoflagellates have left some imprint. Diatoms have left also some imprint as well. And coccolithophores are perhaps the one that have left the best and the most well-preserved fossil record. In that sense, is this aspect making them good models to answer a question of about speciation? We have also a motivation to do that in the era in the post-genomic era. We have access to so much data with genetics that we can answer much more questions than just a question of speciation. We can also assess adaptation and predict how some populations will evolve through the current climate change. And definitely with the environmental change induced by human activities, there is a big challenge in this. So if we look now at the diversity of the eukaryotes, phytoplanktons are quite diverse for sure, and they are spread along that tree. So we can see with green arrows or circles some main groups where we can find phytoplanktons. And the haptophytes in here, where the coccolithophores are belonging, are still some part of a branch that is still not well resolved. So definitely, it's not a failure of molecular biology. It's just an unfinished business where we still have to improve our ways to assess the phylogeny of those organisms. What is clearly what is clear is the phylogenetic signal of those, the deep phylogenetic signal for those organisms have perhaps been lost through really strong pressure selection in the past. If we look at the haptophyte cells, it's again another pattern of diversity where we have different shapes of scales, structure, different flagella. So the main features that is common for all haptophytes in terms of structure is the haptonema, which is another appendice that is not structured like the flagella. It is quite unique in the living world. And yes, and the phylogeny of haptophytes had been, let's say, mentioned as resolved in the early age of phylogenetic analysis. It's not really true, in fact. There are still some unsupported branches, and those unsupported branches are quite troublesome in a way, because they are the one that are, some of those branches are the one that we need for calibration of the haptophyte molecular tree or the molecular clock. It doesn't mean that the calcihaptophicidae is not monophyletic. It's just not always supported depending on which genes we are using when we are making a phylogeny. Again, it's not a failure. It's just an unfinished business. So if we look now inside the calcihaptophicidae, we have around 280 extant morphospecies that have been well described. Some of those species have been recognized with morphotypes where there are subtle variations on the formation of the lids. Most of the species are distributed in between the tropics in low latitudes. And if we look now at some examples of morphospecies with morphotypes, we have Umbilicosphaera we have Umbilicosphaera, we have Helicosphaera as well. The Cyanocosphaera is another case. Calcidiscus, Coccolithus. So one question that was asked in the past with Marcus Geisen, Jeremy, and Alberto as well, Alberto says, was the question of the significance, the biological significance of these variations. So they try to answer the question of those variations with a single gene approach at that time, which was the most advanced method at that time. And in a way, they resolved quite a lot of incongruency, or let's say, instead of having morphotypes that are just subspecies, they clearly show that the genetic variation between those morphotypes can be more attributed to species, actually. So it was definitely the first study providing compelling results on cryptic speciation in phytoplankton. It was reconciling morphological variability with genetic divergence. The luck in that work was mostly due to the fact that those speciation events were corresponding to deep divergence. 1 million year, 2 million year is not 300,000 years. So this study proved that coccolithophore could be a good model to study speciation. And I guess with that affirmation, thank you for your attention. I think I finished my talk. Oh, but in fact, I think I made a mistake. No. Here, species have been established a while ago, as I mentioned, but the study is not really assessing any speciation process, I think. Yeah, the study was more describing the fact that species with subtle variation can be can have a wider genetic divergence. So it's not exactly the same as studying the speciation of a species. And it was mostly relying on the phylogenetic species concept and the morphological species concept. There were no assessment of the reproductive isolation. The paper was not talking much about different habitats, so it wasn't talking a lot about the ecology of those organisms. And the fact that the genotypic cluster concept was not assessed as well is not a big issue, but it could have been helpful to at least estimate what is going on in terms of biological species concept, because we are separating those concepts, but they are all linked in a way. Again, it's not a failure. It's just an unfinished business due to the limitation of the technology at that time. So we can try the same approach with something a bit more recent, at least in terms of recentness of speciation, but also in terms of recentness of methodology. So if we look at the isochrysidalis, we have that famous species, Emiliania huxleyi, that is really widespread. It is ecologically successful, and it has been for many researchers exposed as the model for phytoplankton, for coccolithophores. So it is belonging to the family of the Neurorhabdaceae, and is closely related to the genus Gephyrocapsa and Reticulaphenestra. If we look a bit closer in time, the Gephyrocapsa arose around 3.5 million years ago, and they diversified, and the Emiliania huxleyi species seems to evolve from it around 290,000 years ago. If we look at the fossil record of those Gephyrocapsa, and if we apply a stratophenetic approach, we can see here another way of looking at the evolution of that group. We can see that through time, that group seems to have evolved or increasing his coccolith size 3 times. So it's something that many groups, it's a pattern that can be seen in many coccolithophores as well, but it has been also well described and well documented in the Reticulaphenestra, which is really close to the Gephyrocapsa. Now, if we want to couple that pattern of fossil variation or phenotypic variation in the fossil record to what we know with the genetics, looking at classical genes that have been used in many organisms to first separate species and study speciation, we can see that we have a strong decoupling between morphotypes and closely related species and the phylogenetic influences. So it's something that haven't been resolved since the early stage of molecular genetics applied on phylogeny. So it is quite overwhelming because it's nearly 30 years of single genes phylogeny dogma applied on this group, and we haven't been able to resolve that issue. So again, it's not a failure; it's an unfinished business. But those results are quite interesting because none of those phylogenies are even supporting each other, as well as they are not supporting any morphospecies inference. But they are all supporting that Emiliania and Gepharocapsa could not be two separated genes from a genetic perspective. And if we are considering structural similarities of the coccoliths between those two genus, Gepharocapsa should be perhaps a more objective choice of name instead of Emiliania huxleyi. So I might remind something that I mentioned quite a lot of time since I've been coming to the INA meeting. Belonging to the same genus doesn't mean that those species are the same. They are still different species. So it seems that with genetics, we have been limited for quite a long time. So some morphological assumptions are fitting partially with genetic analysis, but clearly with this group, we have a problem, something that was not seen with the other groups I mentioned in the beginning. So what could be the closest phylogeny we can reconstruct to the real evolutionary relationship between the Gepharocapsa? So that's something that can be potentially reachable if we are approaching that phylogeny with in-depth methods, such as using as many markers as possible. So that's something that can be attainable using different techniques, like it can be transcriptomics or genomics, for example, to mention two. The main outcomes by using those methods is, first, we may have a species tree instead of having confusing gene trees as we had in the previous slide. We can also assess a measure of reproductive isolation between the defined populations through time. So it will be based on a mixture of a genotypic concept and an approximation of a biological concept. Because the genotypic concept is relying on the analysis of population and the frequency of the alleles. So when I'm talking about alleles, it's the frequency of genetic diversity in a way within those population or and between the population. And the departure from shared patterns of variation, so the departure from variation that are shared, normally would correspond to the divergence. So it will be a clear signal of species separation. So in a way, reconstructing the phylogeny can be achieved with genomics, and at the same time, we can approximate the biological species concept. For unveiling the mechanism of calcification, that will be for another talk, perhaps in 1 or 2 years. So I may remind that the Emiliania species, or Gepharocapsa huxleyi in itself, is quite recent. And for Gepharocapsa oceanica, there was a doubt on the dating of its appearance due to the pattern I mentioned before. Now, to assess the genomes of those organisms, first we need strains and a reference genome to have, in a way, a reference on which we can compare variations. And that reference genome was published in 2013 by Betsy Reed et al. So it's a 150 million base pair. So it's a bit like the genome of the Drosophila fly. So it's not considered as a big genome, but it's a big genome for the size of the cell. Unfortunately, the assembly is quite bad. It's 7,000 contigs, so it means that the genome assembly, instead of having chromosomes like we have now for humans, humans have 23 chromosomes or so. Here we have 7,000 small bits to work with. So it's a very fragmented genomes. For the type of work we are dealing with in terms of assessing speciation, it's not big. But for finding the mechanism of speciation, it can be a more problematic job. And we have already access to 12 other strains for which the genomes have been sequenced. The technology we use is quite simple in a way on this scheme. We extract the DNA from the culture using classical physical extraction. So we just use phenylchloroform as chemical, and those chemicals are in fact retaining the DNA on the upper face and all the other molecules on the other face. So it's more a mechanical extraction in a way. And once we have that DNA purified, we need to cut that DNA in small bits, and then we prepare the libraries that is shown here. So we have different adapters that are put on the different fragment of DNA. The different fragments of DNA are then amplified and sequenced, and then we get what we call the reads. Once we get the reads, we may use here a reference genome. So that part is more in silico, of course. The reads are the small sequences, the small sentences representing the genomes. And what we want to do, is reconstructing. Instead of working with small sentences, we want to get larger sentences to match with the reference genomes. And by matching with the reference genome, we may have some places where we have variations, and those variations are, let's say, the most represented variant for different individuals in the population we are working with. Oops. And from those variations, we can then work and, in a way, compare the different populations, compare the different individuals. As a first study, we worked on 10 strains of the Jeffrey-Capsa complex that were encompassing quite a large diversity of the group. And they were coming from different parts of the world. So we reconstruct the phylogeny for those 10 strains. So one way to do that is by aligning all the reads and then do like for a single gene approach, run a phylogeny, or we can cut again in silico different parts of the genomes and reconstruct that phylogeny first by reconstructing different trees as represented here. Here is a superposition of different phylogenetic trees. And here is a consensus tree of all those trees. So we may have something like 1,000 trees here superposed, and here it's a statistical approach made to simplify that view. So it's quite messy in this tree, as you can see. It means that there are a lot of incongruency, as we have seen before when I have shown the study using single gene phylogenies. What is quite interesting with this tree is the fact that we can definitely separate the different morphospecies. They are fitting with the phylogeny. The fact that we have quite a lot of incongruency here is also based on the fact that a lot of things have occurred during those time. So it mostly due to variation of population size. And that's something that measure of genetic diversity can more or less estimate with the allele frequency methods. But we can also, and then we can get those, not really measurement, but those relative size of the population, of ancestral size, of ancestral population size. It's not true population size, it's effective population size. The effective population size is the population that can provide offspring in a way. So they are the most efficient at keeping the species reproducing. And if we look at the excess of shared allele, we can measure what we call the gene flow. And the gene flow is definitely, in this case, how species were still reproducing between them. So in a way, where we're seeing that graph here with the arrows here, we have some gene exchange. So we have instance of reproduction. But the numbers are quite small. They are not in percentage. There are more numbers representing the number of individuals that have provided genes from one species or from one population to another. So what we have seen is in most organisms, when we were using genomic approach, the reproductive isolation is never 100% between species. It's quite interesting. It's challenging, actually, the biological species concept. But it's so insignificant that we can definitely still talk about species. If we integrate that phylogenetic tree on the fossil record, then things are making more sense. And if we look much closer into the subtleties of those coccolith of the G4 capsa within those size increase, we have, in fact, instead of having one G4 capsa oceanica or whatever the name that have been given since, let's say, 1.3 million years, we have perhaps more than two species, more than one species. So we have again a pattern of cryptic speciation that have been recurrent in at least 3 stages. And those 3 stages here are associated with increase in size of the coccolith, and definitely having the morphology that is fitting with some part of the genetics. If we increase the data set for the the Jeffrey capsa, like adding Reticulaphilestra sessilis in our datasets. So it seems that it's not Reticulaphilestra sessilis is not far from the Jeffrey capsa in general. So perhaps it's not a Reticulaphilestra. So maybe we may have to change that as well. And it doesn't change much the pattern, just that we can now see more clearly as well in Jeffrey capsa axillii. We can separate the A morphotype and the B morphotype. That's something that I will present later on one of these days when things will be thoroughly or more in-depth analyzed. But by adding more strains, we can definitely assess more speciation models. So in this study with Dimitri, we tested different speciation models based on patterns of frequency variations within the genomes. We tried 4 classes of speciation, let's say of model. So the first model is the strict isolation. So we start with an ancestral population or an ancestral species, and that species split in 2. And during the evolution of the 2 species, during the speciation, there is no gene flow, so no exchange of gene between the different populations. There is another model that is isolation with migration. So migration is another term for gene flow. We say migration because we are talking about the number of individuals that are exchanging genes between populations. And the isolation with migration is a continuous pattern of speciation where you have a continuous gene flow. And we can have 2 other let's say, specific models still based on the isolation with migration. We can have the ancient migration or the secondary migration. So the ancient migration is when the exchange between the population is still close to the splitting event. And in the secondary contact, it's when the gene flow is way more recent. We can also link those models to the classic mode of speciation I've been describing before in terms of spatial implication. So we have the strict isolation that is clearly linked to the allopathic model. Then the secondary contact more as well to the allopathic model mode, but can also overlap with the peripatric, and then the same with the ancient migration model. It can be in between the peripatric and the parapatric. It can also touch a bit of the sympatric and the ancestral. The isolation with migration is clearly the extreme of that gradient of speciation. Now, if we compare all the pairs of species we have in this tree, we found out that most of the models that are fitting with the variation frequency of is a secondary contact. Then we can definitely deduce from there that most of the speciation mode of those coccolithophore is between the allopatric model and the peripatric model. And it's quite interesting because it's a bit like what is happening to many terrestrial animals. We can attribute that speciation pattern to the formation of barriers in the ocean. As I mentioned before, we can deduce that there is some structure in the ocean. And it's even more obvious when we look at sea surface temperature, for example, where we have different fronts. And interestingly, most of the species we are dealing with, or even coccolithophores, are also distributed according to those structures. And those barriers might have likely changed during the Ice Age oscillation. So to conclude on this work, we definitely have a good match between morphospecies and phylogenetic inference when we are using genomes. And it's providing really good resolution on the mode of speciation in open ocean for cosmopolitan coccolithophores. Now there are quite a lot of remaining questions. I'm just putting some of them here. How does this speciation more translate in the fossil record? I have to admit that we may speculate on that at the moment. But there is a PhD student, Odysseas Arkontikis, who will try to answer that question. He will also try to find some implication, biogeochemical implication on those patterns. Now, will we find the same process in other species of phytoplankton? Well, one way to know is to try. At the moment, it's quite difficult because there is not a lot of fossil record for most of phytoplankton species. However, the fossil record is interesting because you can calibrate your phylogenetic assessment on that, on the first occurrence of a species. However, there is another way to calibrate evolutionary rates. So one way to do that is just measuring the the emergence of new mutation in the background of a strain. So through time, we are always accumulating mutations, and there is a simple way to measure that. You just grow your strain in low density, and through time, like after 1 year, or I would say a long amount of generation, you might be able to get you can sequence the genomes of those different lineages you will create through that, let's say, long-term experiment. You don't expose your strains to different conditions because you're not looking at the adaptation. You're just looking at how spontaneously the mutation are appearing in your strains. And through that new comparative genomic, you might be able to measure the mutation rates of your species or your strain. And from that mutation rates, then you can calibrate a lot of analysis, especially ratio analysis like the speciation analysis I mentioned before. So it can be either calibrated with fossils or with a mutation rate that had been estimated in the lab. And now one question that I'm really fan of is how diversity emerged in the successful Jeffrey Kapsak sclerae. So we, in the nanoplankton community, I guess we all know a bit about that species. It's quite diverse. and it's coming soon. So coccolithophore are good models to study speciation, and it's mostly due to the fact that they have the most well-preserved fossil record among the phytoplankton. Many species are easy to isolate and maintain in culture, and for most of those species in culture, the morphological analyses are well established, as well as for the fossils that are related to those species. So there are still constant developments and optimization, but clearly it's something that is working much better than many other species in the phytoplankton realm. And with the technique we are using for that kind of study, the genomic complexity can easily be reduced to assess the variability. It works really well when we have a good reference genome. So I may thank all my collaborators on those projects. So it's Ross Rikabi, Dimitri Filatov, Odysseus, Arkontikis, Kyoko Ageno, Ian Probert, and Jeremy Young. And I may also thank my team, the Ocean Bug, for the inspiration and the motivation. Well, thanks a lot for your attention, and happy Earth Day.
Thank you, Mahdi. Thank you very much for this really, really brilliant talk. So, does anybody have a question? I'll check also YouTube. No. Any hand raised? Yeah. There is friend of dinosaurs that wish to talk. Jeremy, your microphone is off. You need to turn on your microphone.
I'm sure other people will come up with questions, but while they're thinking, Mahdi, yeah, that was a great talk. Really interesting to. I know we've been working together. It's really interesting to see you actually put the talk together as well. One of the aspects which really intrigues me is in your, as I understand your model, the idea is we get the separation occurs because of ecology. It's some way they're reproductively isolated, and then you get then we can get a genetic differentiation. Yeah. Well, I don't. But if that was the case, if that's an accidental, there's an ecological separation into niches, or there's a biogeographic isolation, then why should the morphological variation so neatly parallel the genetic variation? If it was just reproductive isolation, then you might expect to have lots of genetic species which didn't have a morphological character. I mean, do you think that's a problem, or am I missing something?
No, you're not missing something. It's a really good question. I guess you have separation of two population into different species. It really depends on what are the pressure on those species, on those populations. Sorry. So likely, if those two populations are separated by, I don't know, especially if they are, let's say, in the tropics where you don't where you have pretty much the same pressure almost everywhere, like high temperature and low nutrients. What will cause, for example, an isolation of species is just the fact that the substrate will be so oligotrophic that especially it will be hard for the two populations to breed or to meet together. That will cause, through time, separation of those two populations. But the fact that they are under the same pressure won't necessarily make them change morphologically.
Yeah.
Then, when you have like strong change, like a strong phenotypic variation, it's definitely where you have different evolutionary pressure. Is it answering the question?
Sort of. I still think it still seems to be that just more correlation between morphology and genetic difference than I would have expected. I expected the two almost to be like occurring independently and separate. And it's, I mean, the alternative is in some way that actually the morphology is more adaptive than we think it possibly can be. And so in some way the morphology differentiates, and that then pushes them into gives them different ecology and separates them.
Yes, the thing is, you have the phenotypic answer is quite immediate. So you may have a rapid plastic answer of a cell, and yeah, clearly you already see morphological variation in one individual or one population.
Yeah.
So that's clear. For me, it's clear the morphology is perhaps moving forward the genetics in a way.
Yeah. Okay. That's basically. But it's quite a curious conclusion. I think it's quite it's a interesting one, and maybe that partially reflects why we can do. Why they're so. Some way the morphology is more adaptive than we sort of expected, and is closer linked to the genes than one might have predicted, and therefore that maybe helps explain why they do work so well for as biostratigraphic markers, and for why the it was a curious thing, actually, the degree to which the morphology seems to follow the genetics on lots of levels.
You have another question? Yeah, you go.
We should let other people to ask questions, of course.
There is no hand raised yet, so you can go for it.
Okay. I was very curious about this thing we were saying, where you do have the evidence of gene mixing between separate species. So when you say there's always some gene mix, so if we took Coccolithus and Emiliania, would you expect to see some evidence of gene mixing, or would that be at that stage it really is 100% separation?
Yeah, in that stage, that would be definitely 100% separation.
But have we really tested whether that? As of course, the other possibility, it could be through something like viral interchanges, or some ways going not through breeding, but some type of genetic exchange which isn't through reproduction.
At the moment, any of those haven't been checked or tried, mostly because the genome is quite difficult to work with. But we will need more genomes to try those aspects.
But I guess, your basic the sort of hypothesis you're suggesting at the moment is that occasionally you would get, very, very occasionally, you would get reproduction between two of the two haploid phases of something like Gephyrocaps and Emiliania would actually end up mating.
Yes, that's the underlying hypothesis.
Oh, well, so maybe we should go back and try some reproduction experiments.
Yeah, we will need to isolate quite a lot of those haploid phase. Yeah. If they are stable.
Okay, I have a question from YouTube. I will read it and also type it just in case my pronunciation is not great. So it says, "Hi, great talk. A lot of coccolithophore species have large genomes, over 150 mbp. I don't know what's mbp, but I mean, I know in French, but I don't know it's pronounced in English. So sequencing takes time and can become quite expensive. Have you found a more suitable locus than the TUF-A18S for routine PCR-based species identification differentiation in your genome analysis?" I wrote it down also, yes. So you may be able to read it again. That's just the first part, actually. And the other part was that. Actually,
Yes.
the bottom one is up normally, and so on.
Yeah. No worries. Yeah, so for MBP, it's a million base pair.
Yeah.
Yes, it's true. Sequencing takes time, and it's for this type of work, it's very expensive because we need a lot of individuals. And I have presented something in the first analysis. I'm presenting 10, and then I'm presenting something like 40. I would say it's not enough for the even if the results are robust. We are having a good statistical inference and are quite reliable, but it's still not enough. There are some species for which we have one strain, some for which we have 2 or 3. So that would be great if we can get more strains for them. But yes, it's increasing the cost of that kind of analysis. But to answer the question of the PCR, the single gene approach. So I have been trying to find that for those last 5 years, and I have to again rely on the dear Master Yoda. I'm sorry, it's not a failure. It's a work in progress, but very little progress because I haven't been able to find something suitable at the moment.
Okay, thank you. There were another question in the chat that time. It says, thank you for your talk, Mahdi. What causes speciation?
That's a really philosophical question.
Okay. If you want to continue, or you stop here because maybe it's
okay. I guess yeah, I might stop here.
Okay. So maybe you can discuss about that. I hope during INA conference. There is Gerard that is actually raising his hand. So Gerard, I'll let you talk then.
Hello, Mahdi. Thanks. There was a brilliant talk. Enjoyed it very much. I've got one question. So, I mean, you put a lot of work into the GHUX group and done all this analysis. So, I suppose not so much in theoretical terms, but more in practical terms. How transferable is that to other species? So, do you think that what you've done on GHUX can be done on another species like Calcidiscus or whatever?
I hope so. So the thing we really don't know what is the amount of intra-specific variation in this species. So we have a view again biased by the morphology and the single gene approach, where we have always one. We try to have one branch for one species, and if we, yes, the main thing is we don't have a lot of those species in culture, so we need again something similar, and variation have to be assessed in a way. I don't think the best approach is starting right away with genomes of all of these guys. We should perhaps start with a really low complexity approach, like using transcriptomes to have access to the genes and then compare, making references and compare those different individuals with a reference. And then when we see variations, then we can start thinking about working with full genomes.
Yeah. Do you expect similar results, like in terms of your overall conclusion and the speciation model?
At the moment, I would say yes. I don't see it can it can change. In fact, that's the thing. It can change. There might be more to it than just allopathic speciation. But at the moment, it's hard to go further due to that limitation I mentioned. The fact that we don't have more
I know that.
individuals.
I just wanted your gut feeling, but I know you can't produce the data now.
No, but with my gut feeling, I really hope that there might be more variation, more model.
Okay, cool. Thank you.
You're welcome.
I guess we can wish you to be able to work on that as much as possible, to have those answers in the future. Is there any other question? Oh, yes, Patricia.
Hi, Mahdi.
Hi.
Hi. Thanks for the great talk. So I have a question. Is more like I would like to know your feeling about more the ecological perspective of your work. So you basically elaborate on the low latitude speciation and specifically in the tropics. And my question was about more the gyres, so the specific feature that you have at this latitude, so the extreme stable conditions that you have in this very oligotrophic parts of the ocean. So are there evidence then in terms of evolutionary pressures and biogeographic, I don't know, paleobiogeography of the ocean, that there is really a relationship between like gyres, extensions, and so on, and speciations in the past? And then the other questions that's not related to this one is about extinctions. So your talk is about speciation, and I was wondering if you can elaborate then on extinctions. It's just like, is the opposite of speciation really, or is something else?
And for the first question, that's I've been interested in that one for quite a long time, because even in small eddies we can see change in the structure of the communities. And if we have change in the structure of communities, it means that we have potentially change in the genetic diversity of a population. So that's something that can be seen in GHUX in different eddies. We can see first a change in distribution depending on how the eddy is working, if it's a downwelling or an upwelling. So it's drastically changing the genetic diversity within. So we can only speculate again on and predict that if we are looking at a big gyre, we may have definitely changes in the genetic diversity of the population, and it might be working as a trap for speciation for one species or two species. I don't know, but it's really hard to answer. It's more speculative in a way. About extinction, it's more difficult to look at this when we are looking at living things. So it really depends on how far we can calibrate our genetic groups, our genetic species to the fossil record. If we can go within, let's say, a population approach, like having one species and having a lot of individuals for that species, maybe that's something that can be feasible if we develop that marker that is mentioned in the question, that is that earlier question, that something we can use for routine PCR-based locus for looking at what is inside the fossil record. Developing a phylogenetic approach can be really useful to assess those extinctions, because we will be able to see to follow the living things, see where they speciate or where they diverge from the main ancestral population. But we'll see also all the divergences that are appearing and disappearing through time. I hope that answers a bit the question.
Sorry, my bad. Is there any other question? Because I'm also preparing something for the end of the talk, for the conclusion. So is there any other question? Okay, there is one here. I'll copy it. Oh, and of course there is a mail at the same time. Most of the work on coccolithophore taxonomy has been done by micropaleontologists based on morpho structural features. Do you think do you think we have done a good job based on your technique?
So
for me, it's morphology and genes are telling a story that's quite similar?
No, it's a really good job. Whatever the job had been done, it's very good. The issue we have with morphology is knowing where we have to stop. And most of the micropaleontologists have been perhaps better than many other paleontologists. They have been sometimes a bit more conservative, but by doing so, it helped a lot in a way, avoiding over-splitting groups and things. Adding genetic now won't change much of the micropaleontology for the old lineages. For the recent one, it can only, in a way, it can only refine things that have been done. It's not, it can correct things, but it's not big things. It's mostly refining. So yes, micropaleontologists have done a really good job.
And I thank you for that. Okay, is there any other comments, questions? Just let me do a quick check on YouTube. There is none on YouTube. Okay, but I guess we can stop the discussion for today here. So I have two things to say to everybody. Firstly, and I guess I saw her, she was there. Yeah, Jessica Walker will be the next on NannoTalks next month on May 16th. And she'll be talking about coccolithophore crystal chemistry and polysaccharides and beyond. That's the title of the talk. And I thank her for accepting the invitation to contribute to NannoTalks. Actually, the one after, in June, will be the last one, and it will be by Catarina Guerrero on paleosonography also. So this is our two last speakers for NannoTalks. After that, there is summer rest for NannoTalks, and then there will be INA. I hope you all received the announcement email, Facebook announce Jeremy on the web site of INA. I can, as an organizer, I can only recommend you to register as early as possible, to do the abstract submission as early as possible. And if you have any question, any issue, you can contact us at ina18@cerej.fr. Don't hesitate. We are 3 behind this web, this email address, and we'll be able to help you. I know that the registration process is quite heavy, but we have the tools that our institution are giving us, so we cannot actually change. That's it. I hope to see all of you at next NannoTalks, and then also at INA, and also Mahdi, you'll be in Canada at that time. But I hope to see you coming back to France for INA.
I will
You
try.
will.
I will try to come back.
That's great. That's great. Oh, you say you will. So now it's set.
I said I will try.
Okay. Okay. Well, okay. I hope you'll be able. And I guess I stop here, and I wish all of you having a good day, afternoon, morning, and so on. Thank you, Mahdi, for your brilliant talk. And I hope we can continue this discussion during next INA.